Hi, we're group 6, and our presentation is on celiac disease. So just for some context on SNPs or SNPs, a SNP is a single nucleotide polymorphism that leads to a change in the genome due to a nucleotide shift, and ultimately this will lead to some form of genetic variation. And so the purpose of this particular project was to analyze the genotype of an anonymous donor and determine any SNPs that could have caused genetic disease. And so the way that we chose our trait was just a personal interest and an interest due to family history. And we narrowed down the RS number with an online article. And this has been heavily studied in UK and US populations. And so our RS number here is the marker associated with celiac disease, and it is located on the first intron of the HLA-DQA1 gene. And the HLA is the human leukocyte antigen that is located on the chromosomal position 6p21. And this is just a gene map of the HLA region.
So this SNP is of interest because it's associated with an increased risk for celiac disease. It's essentially a very advanced chronic gluten intolerance, which causes a lot of gastrointestinal discomfort after you ingest any grains with gluten. It's basically an autoimmune disease. It results in inflammation, and if you can see here in figure 1, the villi of the small intestines starts to become degraded. So in the short term, this is going to cause low nutrient absorption, some diarrhea, abdominal discomfort, and the swelling of these intestines. In the long term, it can lead to much greater things like anemia, osteoporosis, and some forms of gastrointestinal cancer. Overall, it's very difficult to diagnose without serology and genetic testing because the symptoms could be of so many other diseases. So it's kind of a last resort kind of diagnosis.
And so the way that the susceptibility works is a G to A mutation on this locus. That means that you are likely to have celiac disease. Homozygous for A is the greatest risk, and heterozygous for AG is some risk. And so the mutation on the HLA locus increases your risk by about 7 times. And literature found that it was more common in females than males, which is about a 2-to-1 ratio. And celiac disease depends on like a multifactorial inheritance. It's not just if there's this mutation, you have the disease. And the HLA region is involved in about 40% of that heritability. And so the mutation itself leads to a heterodimer in the HLA-DQA1 gene, and that contributes to the susceptibility of it. And basically, it creates an antigen that binds CD4 and T cells and triggers that immune response, hence why it's an immunocompromised individual. And over here, we just have a map of the HLA complex on chromosome 6, and this is a class 2 DQ region on the HLA-DQA1 gene.
The process of finding a good SNP to research is very tedious. I recommend first searching on Google Scholar for diseases caused by SNPs. Once you locate an interesting article, make sure it has an RS value, and then you can paste this into both of the genome data sets, which are anonymous, just to make sure that it's even data that you can analyze. If it's not present in either one, you should go find another SNP. And after you find a good one, you should verify the RS number on OMIM. You can click it to find more information and links to other studies. And then afterwards, you should paste it into SNPedia to verify that both GenoMag summary and population statistic tables are available. And if one of these tables is missing, I would recommend choosing another SNP.
All right, so going into the results of what we found. So if you look on the table to the far left, this shows our SNP of interest, which we found in anonymous data set number 2, which was provided by our instructor. And the SNP was found on chromosome 6, and it was one of over 600,000 different SNPs that were sequenced and analyzed. But as you can see, this individual happens to be homozygous with a genotype of CC. And so this corresponds to a genotype of GG on complementary strands, which is a little more relevant to the table in the middle. So this figure provides a summary of the genotypes and phenotypes associated with this SNP. So being homozygous AA poses an increased risk of celiac disease, being heterozygous is a somewhat increased risk, and then being homozygous GG is average, meaning a reduced risk for celiac disease. And then the last table on the far right just shows how the genotypes associated with celiac disease vary among different human populations. And you can see that the blue bar, which corresponds to a genotype of GG, that is the most prevalent across all of the populations. So to go into the interpretation, like I mentioned, our sample demonstrated a genotype of GG in the location of interest. And so their predictive phenotype would be average or a reduced genetic risk for celiac disease. And it was found that the odds ratio for developing disease was 0.3 in a study of about 800 patients who had this genotype. And so in terms of predicting a race, one potential predicted race is Japanese ancestry, and that's because this population demonstrated the highest incidence of the normal or GG genotype. And of course, all of the populations had that genotype being most prevalent, and so this is just a prediction that we made. Lastly, based on the data, which showed data from both the X and Y sex chromosomes, we determined that subject number 2 was a biological male. And just to wrap up with some of our reflections on the project, it was a really enriching experience because we got to learn more about how to interpret genotype data sets. And even though we kind of knew about SNPs from prior courses, this helped showed us how we can correlate them with genetic disease and use interesting resources like OMIM to simplify data that seemed initially complex. We're excited to apply these skills in the future and potentially assess our own risk for genetic disease to make more informed decisions about our health and well-being. And these are our sources. Thank you so much.